So first of all, I thank the organizers of this conference and especially Dr. Nandini Rajamani for taking the trouble of inviting me to this. It has been a very interesting meet, and I'm very sad that I could not actually come in person to attend it, but I attended whatever I could online yesterday, and it was really very nice to listen to all the keynote speakers and all the others, including Dr. Mathur. So it's a great pleasure for me to talk to you in this symposium. And as it says here, I'm going to be talking about how we use pollen to reconstruct the past vegetation. And I'm going to present it with a few case studies from Southeast India. And one of them is actually from Andhra Pradesh, and the other one is from Tamil Nadu, but very close to Pondicherry. So there are two co-authors in this presentation, which is my colleague Prasad and also Dr. Navya Regu, my former PhD student, who is presently a postdoctoral fellow at Manipal and an associated researcher at IFT. So we'll start with definitions. So what is paleoecology? So paleoecology is very simply the ecology of the past, and it deals with the reconstruction and study of past ecosystems, including relationships between organisms and their environment. But we don't have the organisms and the environment, do we? So we need to use proxies. And to look at the environment is always a continuum. So this we'll have to keep in mind when we go about this definition. So you could also say that it's a branch of ecology studying past ecological systems, their temporal dynamics, using fossils and other proxies. But it's also the past of the ecological systems, in a sense. So these definitions you can know a lot more from Valentin Rull and Burks, who have done quite an exhaustive papers on definitions of this field. So what are the proxies in paleoecology? What is a proxy? Any indirect indicator of what is the past environment. This indirect indicator, as I had also hinted in the previous slide where we defined, it's biotic, abiotic, and all the interactions which are therein. And what are these proxies actually? So it could be pollen, which is what I'm going to be focusing on in this presentation. But equally on the pollen slide, we find charcoal. But we won't find phytoliths because it's kind of a complementary proxy, and I won't speak much about it, but it's also a very important and powerful proxy, especially when you're dealing with, say, archaeological sites, or if you want to understand what has been the past of domesticating crops and issues like that. So a whole number of things, from tree rings to stable carbon isotopes to bones, teeth, and then other micro proxies, which are like diatoms, chironomids, and of course plant macrofossils. But many of these are not as well preserved, and they are not preserved in as many quantities as pollen actually is. So we need to use what we call as a multiple proxy approach, which means that you can do a reconstruction with pollen, but it would also be good to, for example, look at what has been the organic matter variation with that, and look at the stable carbon isotopes. So how does the stable carbon isotope variation match with the pollen? Because that would give us a better understanding. So it's a little bit like a hologram. So you use one proxy and you do get a reconstruction, quite a robust reconstruction. But if you want something which is better resolved, more finely resolved, then the more proxies, your reconstruction is going to be so much more robust. So what we are trying to see is the variability, the response to environment change, and other things, which including resilience, because the past data will tell us what was the kind of vegetation spectrum in the past, and what is it at present, and then we can look at taxa which have been present through a long time and giving this very long baseline of what could be very useful for when you go around to actually defining sustainability. So palynology here deals with mainly the identification and analysis of pollen grains. But the stress here is on the morphology of the pollen. It is based on the wall features. So in palynology, especially, we in fact remove the cell contents, which means that we don't deal with the DNA. So usually pollen studies, people will think about DNA, but that comes especially in the sediment context in a different way. So we could look at ancient DNA because the process that we use to prepare reference slides, removing the cell contents, is what happens in nature also when pollen grains get deposited in sediments. So their inner cell contents get exuded, and it's only the wall of the pollen which is very resistant to decay and which also has all the morphological features which will allow us to identify the pollen grains to a taxonomic group, which is not always species, but we'll get to that later. So why do we actually study pollen grains? Yes, because you can identify source plants with the pollen, and in the sediments, or sometimes if you're looking at honey, or if you're looking at loads which are carried by a bee or any other insect, the pollen groups can characterize what are the plants. So when the plant is no longer there, as in the past, or when you don't know where the pollinator has gone to collect the pollen it needs, you can use the pollen extracted from there, understand it, and then get back to the plants. As I was already telling, it's produced and you can also retrieve it in large number of quantities. And one of the reasons why this is such a good proxy is that when most other vegetal fragments have decayed, pollen survive for a long time. So these are really good tools for identifying plants. And as you all know very well, and I don't have to tell you this, pollen is meant for dispersion. So biologically, it's something which is meant for dispersal. So it is produced in large quantities, it is well dispersed, and also because of its wall properties, it's quite resilient to lab treatments. So this is why pollen grains are very important in various studies, including in reconstructing the past vegetation history. But there's always caveats when you use any method. As I said already, you can't identify plants at the species level. And pollen preservation, of course, generally it's very good when it is not, when the conditions are not arid, when you have non-oxidizing conditions, when you have material like clay, so you can have good pollen preservation. You can't have good pollen preservation. So you need to think, as I already said, of complementary proxies. So here, phytoliths may be useful. And it's also true that all pollen are not equally well preserved. So some may particularly not stand the chemical treatment and hence not be represented in the pollen spectrum. So these are some caveats which we need to keep in mind. But then I'll go on to telling you what are the features which we actually observe in the pollen. This is going to be really very quick. So one of the important features is size, of course, as you do with most microscopic objects. But then the pollen also contain apertures, which are called pores and which are also called colpi. So this round aperture is called a pore, and this elongated aperture is called a sulcus, and it's called, and the wall ornamentation itself can have some patterns like reticulation and striation. So what I'm going to do now is actually to show you how it looks, under the microscope. So this is a pollen grain of Sida, and when we look at it under the microscope, Um, so I hope you can see the video playing. So basically the light microscopy is used in the same way that you use for sectioning plants. So we see a yellow pattern. So you start with the top of the grain, because within the microscopes, within the slide, this is actually a 3D object. So this is a pollen of Sida, and this one is one of Manilkara. So you see the patterns, and you see the exine ornamentation here. You can see the long corpus here, and you can see also the pore. And you can also see that there are actually 4 of them. So you know the symmetry, that it is what we call as 4 colporate. But this is what a fossil slide looks like. So the pictures, the videos I showed you, they were from our pollen reference collection, where we really want to know and study the morphology. But this is actually what this looks under 160 times magnification, and this is 400 times magnification. And here's one pollen which you can see. So it's not that great, but then normally we start training, and we train students by using pollen from honey, which is like really very rich and diverse. So you can see at least 10 different types of pollen grains over here. And this one is what we call as composite akinate, but it could be many things. It could be blumia, it could be sunflower, it could be a lot of things. And this, on the other hand, is belonging to Feltophorum, because that's quite a favorite of the bees. And then how do we collect the sediment samples? So this is just an illustration of one of the corers that we have been using. This is a modified Russian corer, and it's manually operated. And these are actually images from one of the field sites in Andhra, where we have collected sediment cores. So the process is to collect the core, to analyze it, and finally to reach to this pollen diagram. And this again is one of the sites that we had identified in the course of a project with the help of both remote sensing, because we had done this jointly with the NRSA at that time, and also the Forest Department of Andhra Pradesh, where we had a lot of help once we identified many sites like this. And this was the other site which we had identified. This one is Rushul Cheruvu in Mehboob Nagar district. The previous one is actually in Rajampet, and it's Ellamarazu Cheruvu. And then finally this one, Kotapuram Cheruvu, which is the one which I'm going to present the results of. This was identified in Lingal RF, and it was sample for pollen. And this is actually the location of the site. So this is Kotapuram Cheruvu, and we got a core out of this, and we also got it radiocarbon dated, and we found that we got a record which was going back from a 1 meter core, which was going back to nearly 1772 AD. So a little before that, probably. And this is what we call as the percentage pollen diagram. So now you see that I've marked a lot of taxa here, and you also see I've written Justitia T, which is the pollen type. So we also have a lot of the forest taxa. You have Hardwickia binata, which is one of the pollen which we can identify actually to the species level. But we also have a mixed group of Melastomataceae, Combretaceae. But in the dry deciduous forest, of course, this is an important component. We have Lania, we have Glaucidion, we also have Schleichera, as well as Dodonia. So what we did was also to quantify. So the other important thing which allows pollen to be used in a very powerful way for past reconstruction is that you can quantify the pollen per gram of sediment, but you can also quantify using modern analogs what is the distance weighted pollen measure and apply it using another modeling approach, which I will come to in the later part. But here, for example, what we saw with the pollen concentration diagram was that when we summed all the forest taxa, there was a slight increase on the top, which was a bit surprising. But you also see that one of the key disturbance markers, which is xanthium, has also increased quite a bit on the top. So which means in the more recent times. So we just took up this particular thing. So one of the conclusions of this work was that though the forest cover remained unchanged, some important species like this Hardwickia binata was not very evident. So even if on the top we had quite a good recovery, we still had quite a good recovery of the pollen types in the recent time. It was not. So we also noticed that there was this practice of loping of Hardwickia binata trees, and though it will not destroy the tree in the short term, it may affect flowering cycle and pollen productivity, and hence the entire reproductive cycle of the this particular plant. So we did this only with reference to this one species, and then we have published this already in Botany International, and then we continued a little bit more analysis looking at the rainfall data. So this is what I meant by saying that it's quantitative data, and we were getting some good results with reference to correlating it with the rainfall variability taken in different cycles of rain adjusted. So all of this is very important, but if we want to apply this pollen data into models, that is climate models, which are using feedbacks from land cover, then we need to go about in a slightly different way. So as I already mentioned in the caveats, there's no one-to-one relation between the pollen and the plant, and also it is only indirectly that the climate is linked with it. So we need mechanistic models to understand pollen vegetation relationships, and these have been developed in Europe and very well applied all over Europe and in many parts of some parts of North America and Canada, and also recently in Africa. So, and of course, my PhD student Navya did this work in Southeast India. So this requires us to do vegetation surveys, which is very, very important, and it needs to be distance weighted. So what do I mean by distance weighted? So you take a pollen sample here, and then you have around 10 meters, we have 21 quadrants of 1 by 1, because the assumption is that the vegetation closest to the pollen sampling makes the maximum contribution. And then we have a zone B, which is also surveyed on the field, and by walking in transects and making these quadrats whenever there is a change in the vegetation. The last part, which is going up to 1 kilometer, it's done with remote sensing. So in addition to this field survey, which we do, and that's Navya there, and also Prasad, we also need the fall speed of pollen. And all this goes into the ERV models, and it gives us an estimate which is called the relative pollen productivity. It also gives us the background pollen and the relevant source area of pollen. But interestingly, what we get out of this work, which is going to be applied to a fossil to the fossil reconstruction, is actually these 1 kilometer radius maps of the vegetation around each of the pollen sampling set. So now we need to have in order for us to get pollen productivity estimates for N taxa, we need to have 2N number of sites. And in her thesis, Navya has worked on 14 sites, and we have got the pollen productivity estimates for about 6 taxa out of that. So it's a minimum of 2N sites. So this is a important side output, because this actually gives us our sampling strategy makes us want to choose sites which are in different degrees of disturbance. So because of that, you get the chance with this to also map the vegetation in a systematic manner in various parts of Southeast India. So this was all done in and around Pondicherry mainly. So one output of this was this paper, which has been published 2 years back. But the other outputs, it's in Navya's thesis, and her papers will be coming out quite soon. So the whole rationale of this extended R-value model is that the pollen loading of a taxa in your sediment record, it's actually the distance weighted plant abundance within what we call as a relevant source area of pollen, which means that you also have a background pollen which can come in because of long distance transport of pollen, and you also have the relevant source area of pollen. So this ERV model, which allows us to get the relative pollen productivities, is then applied to what is called as a model which estimates the regional vegetation abundance from what we call as large sites. So why is all of this important? Because this is the data which actually links what is happening to the feedback from vegetation, which has now become very important in all the climate models. So as you know very well, it's the atmospheric composition and climate which decides what the terrestrial ecosystems are. But then you also have humans interacting with these ecosystems, and you also have the ecosystems themselves providing the cover and giving a feedback. So you have forcing because of the humans and also because of the climate, which is interlinked. And you also have the, again, you have feedbacks from the atmospheric system, which has been so modified back into the terrestrial ecosystems, and so on and so forth. So one of the highest priorities of climate modeling community is to develop these dynamic vegetation models, and these have been done now, and the latest climate models are all incorporating the feedback from land cover, which is feedback from the actual vegetation on the earth, which had been ignored in previous editions of these models. And more recently, the pollen-based reconstructions themselves have been put into it, but of course we are not yet there for the Indian part. But that's why this is very important, because this can be used in climate modeling, and it can be used also in conjunction with the information from land use, actual land use, which we need to interact with historians. Dr. Mathur had mentioned, so there is no escape with interacting with the social sciences and with people who are doing that. And history is really important, and we have also done that in the course of our work, and archaeology, because to understand the past, you need to know a little bit also from the humanities side, how they do the reconstruction. So now going back to the site where we have done the reconstruction in Southeast India. So this is a site which is called Chinnababu Samudram. It's very close to, it's in Virupuram district, and it's very close to Pondicherry. And here we got a 1.3 meter trench. We were not able to core here. And this trench dated back to nearly 6,000 years. So this was a very great thing. And we also got the pollen record, similar to the record that we got in Pottapuram Cheru, but we did all of this work of quantifying the distance-based pollen weightage and getting the relative pollen productivities for some of the taxa. And so we are able to go from the pollen data to the actual land cover. So what you see, for example, is that Acacia albizia is actually underrepresented in the record. So this is because of various reasons, because of its productivity, because of its dispersal, because of various other reasons. On the other hand, you see that Justicia is almost always overrepresented. And you also have another example where you have Amaranthesi, which is again underrepresented. So this is what we mean. So you actually get these values. These are actual estimates of the actual land cover as opposed to just the pollen percentage diagram. So we use palynology as the central tool. We link with people doing geology and geochronology and archaeology, of course, but we also link with modern ecology, because recently we have also started looking at pollen loads from bees and how you can get the foraging preferences of these very important creatures. So we started doing it with honeybees because it's the easiest for us to start doing that, but it could also be done with other bees and other pollinators which actually gather pollen. And I just leave you with these two things. One is from Abayar, which says, because it's a mixed audience, I'm not going to say the Tamil one, it's there. What we know is just a handful of earth, and what we don't know is as huge as this world. And for us, because we use something like the pollen to see a world in the grain of sand and heaven in a wildflower and hold infinity in the palm of your hand and eternity in an arm. Thank you all very much for listening to me. And I'm most happy to answer any questions that you may have now or later. Thank you.